Good morning. Today we are going to study about the histology of central nervous system. In this histology, we will study how the cerebrum, cerebellum and the spinal cord appears under microscope. This is the spinal cord. The spinal cord can be seen in a section. It can be seen like a combination of white and grey matter. In spinal cord, the grey matter is presenting inside and the white matter is present outside. This is grey matter portion and this is white matter portion. It is somewhat like a butterfly shaped structure. The grey matter is in the shape of an H. So this is how the section of spinal cord appears. Now we will learn what are the features present in a cut section. The spinal cord, this is the anterior part of spinal cord and this one is posterior part of spinal cord. The spinal cord contains this major portion, white matter, and this H-shaped portion, the grey matter. The spinal cord can be divided into right and left half by means of an anterior deep median fissure and a posterior median sulcus. The right and left half can be again divisible with the posterior column or the posterior funiculus, anterior column or the anterior funiculus or fasciculus, the lateral column or the lateral fasciculus. It can be seen on either side, the anterior, posterior compartments with lateral compartment. So, like that, in case of grey matter also, the posterior extending part that is named as the dorsal column or the posterior column or the posterior horn. The anterior extension towards the anterior column is named as the anterior or the ventral fasciculus or the ventral column or the ventral horn. At some regions of cut section like thoracic part and the first two lumbar aspects, there is an additional extension of grey matter can be seen towards laterally, that is named as the lateral column of grey matter. So these are the parts which can be seen in a spinal cord when we are taking a section. Through the central part of grey matter column or the grey column, there we can see the central canal through which the CSF passes. So these are the features which have to study in a section of, these are the features which can be seen in a section of spinal cord. The characteristic specialities, that is the tissues are arranged at the central portion as grey matter, at the outer portion as white matter. The grey matter, as we all know, the grey matter is contains with the cell bodies of neurons and the supporting cells also present in it. They are the neuroglial cells, the supporting cells and the neurons, along with some of the unmyelinated dendrites also can be seen in the grey matter. The specialized type of cells which can be seen in it on the anterior column or the ventral column is bipolar neurons. Bipolar means with two processes. The white matter do not contain any cell bodies, only myelinated nerve fibers present at the white matter. The central region of spinal cord is with gray matter and surrounding region can be seen with the white matter. So these are the features which can be seen in a cut section. The same thing, it is all the level. The gray matter is contains with anterior and posterior horns. The thoracic and upper two lumbar segments only shows the lateral horns. And the lateral horns, they are having sympathetic nuclei. Entire gray matter is made up of cell bodies of the nuclei. The gray matter is majorly composed of nerve cell bodies and non-myelinated dendrites along with neuroglial cells. These are the components, three components which can be seen in a gray matter. And white matter can be seen in dorsal, lateral, ventral columns or anterior, posterior and lateral columns. The white matter contains majorly bundles of myelinated and some of the unmyelinated fibers. All these are axons only, bundles of myelinated axons and unmyelinated or non-myelinated axons. It also contains neuroglial cell bodies of some specialized cells named as oligodendrocytes. They are responsible for the production of myelin in central nervous system. So their cell bodies also can be seen in the white matter. And few blood capillaries may be seen. And in white matter, usually nerve cell bodies and dendrites are absent. Majorly, white matter contains the long, elongated, myelinated axons. So this is a diagram how we can draw the histological slide of spinal cord. These are the white matter containing myelinated axons. This is posterior column. This is anterior column. This is lateral column. Here in gray matter we can see the dorsal or the posterior horn, ventral or the anterior horn. Here in ventral horn we can draw the multipolar neurons, bipolar or the multipolar neurons we can draw. And there are the small glial cells, neuroglial cells you can draw here. And so this is the gray matter containing. And this is the central canal. The lining cells are the ependymal cells, that is being the fluid present inside. The lining epithelium is the ependyma. It's a central canal. So these are the features which have to draw in a histology slide. So you can draw the diagram of spinal cord from this slide. So this is a specialized technique of staining to show the lighter regions darker and darker regions into lighter aspects. So in this diagram we can identify the one as posterior median septum, the 7 as the central canal within the central portion of the gray matter, and 8 it is the anterior median fissure. The second one is the posterior white column, 6 is the anterior white column, 4 is the lateral white column, 3 is the dorsal horn or the posterior horn of gray matter, 5 is the anterior horn of gray matter. So these are the features which can be identified. It is very simple to identify, and this is the diagram how it appears under the microscope. The original view under the microscope. All the other diagrams that we have studied is schematic representation. This is how it appears under microscope. This is white column. This is gray matter with posterior and anterior horns. These cells represents the glial cells and the cell bodies of the neurons. The multipolar and bipolar neurons can be seen here. This is anterior and lateral horns. This is posterior horn. So this is anterior median fissure. This is posterior median sulcus. And this is the central canal with ependymal lining. So that is all about the spinal cord. Spinal cord, in a section we can see the grey matter and the white matter with its parts. The next slide is about cerebellum. Like the cerebellum also contains outer aspects of cortical region with grey matter and inner core of white matter. And like basal ganglia in cerebrum, here is also the intracerebellar nuclei. The nuclei, the grey matter connections. Grey matter collections can be seen inside the white matter. So these are the features. So for the histology, we will study the cortical region. The cortex, which are the layers, can be seen at the cortex. That is the histology of cerebellum. The cerebellar cortex can be seen in a section like this. The outer covering pia matter. Just inside that we can see it is divisible into 3 layers. This is outer layer, then middle layer can be seen as a single line, and inner layer it is given as a darker region. Then innermost core is forming as a white matter. So this is white matter region. Then rest of the 3 layers. This is inner layer, this is middle intermediate layer, and this is the outer layers. These are the 3 layers can be seen in the cerebellar cortex. The 3 layers are named as outer molecular layer. Outer layer is molecular layer. Intermediate layer is specialized type of cells named as Purkinje cell layer. The innermost layer is the granular layer. So these are the layers which can be seen in a cerebellar cortex. Molecular layer, Purkinje cell layer, and granular layer. Each this section, that is the surface of cerebellum, can be seen in different leaf-like structures like this, a single leaf-like structures. That leaf-like structures are named as folium. Each folium contains this different 4 components like innermost core of white matter, outer to white matter there is glial granular layer, and an intermediate Purkinje cell layer, and molecular layer. The cerebellar cortex is composed of the cells. The molecular layer cells. Molecular layer is formed with the few neurons, only some of the neurons. That's why it is somewhat like in a spacious or diffused type of neurons can be seen with unmyelinated fibers. So outer molecular layer is somewhat like a diffused with few neurons and unmyelinated fibers. The intermediate layer, this is the Purkinje cell. Purkinje cell possesses a large flask-shaped body with well elaborated dendrites. The dendrites are presenting towards the molecular cell layer, and the Purkinje cell bodies of the Purkinje cell, it is presenting itself at the intermediate layer. So this is a single layer of. So the Purkinje cells are arranged between the granular layer and molecular layer as a single line. A single layer of Purkinje cell layer can be seen, and inner granular layer, they are formed of small condensed form of or densely packed smallest neurons, composed of a densely packed smallest neurons. Small neuronal cells can be seen in it. They connect with the afferent fibers of the rest of the nuclear layers. This is a single Purkinje cell, how it appears. It possesses neuronal bodies with number of dendrites. The dendrites passes or occupies in the molecular layer along with the other fibers, and axons, they are passing towards the granular layer. Through the granular layer, the axon traverses towards the inner core of white matter, and within the white matter it reaches towards the intracerebellar nuclei present in the white matter. So this is the diagram how we can draw the cerebellum. With folia, we can see the cerebellum with folia. These are the each segments, which is a folium. Each folium is lined with a pia matter. Here the 4 is labeled as pia matter. Then inside that you can see the molecular layer with diffused type of neurons. Then next intermediate layer you can draw the Purkinje cell with dendrites towards the molecular layer. Next, concentrated densely packed granular cell layer, that is labeled as the 3. Then inner core of white matter. Each section contains inner core of white matter. So this is the diagram how we can draw in the cerebellum section. This is the microscopic view of a folium. This is outer molecular layer. This dots, large dots representing the cell bodies of Purkinje cell layer. Inner to that we can see the the granular cell layer and inner core of white matter. So that is all about the cerebellum, how we can identify under microscope with the 3 layers and inner core of white matter. Now the cerebellum. The cerebellum, this is outer grey matter region and this is inner white matter region, and this is also grey matter collection. Basal ganglion and thalamus, diencephalon, all these are the grey matter collections. So this outer cortical region or the cerebral cortex can be seen under microscope like this. The cerebral cortex contains a number of different types of neuronal cells. Majorly it contains a special type of cell which is named as the pyramidal shaped cell. Pyramidal cells. The pyramidal cells with its efferents and afferents. The efferent is presenting towards white matter and afferents or the dendrites, they are passes towards external aspect. They are small cells and most superficial. That means it is presenting towards the external aspect. Then next type of cell is the stellate or star-shaped cells. The star-shaped cells can be seen. They are granulated. The cytoplasm is granulated, so that granule cells, it is otherwise named as granule cells. It have got connection to nearest that is the dendrites connecting with the nearest pyramidal cells, and the axon passes towards forming white matter. That is second type of cell. The another type of cell which is also can be seen superficially, that is the cells of Martinotti. The Martinotti cells. The Martinotti cells, they are possessing, or they have got small processes. These processes are connected towards that is dendrites. They are connecting towards the cells, and the axons, they are forming the superficial layer, or they are passing. Usually most of the cells, the axons passes to form the white matter, but here in case of Martinotti cell, the axon passes towards the peripheral region. This is also pyramidal cell layer. There is another type of cell without a typical shape, that is named as the fusiform cells. Mostly it can be seen in a spindle shaped cell, fusiform. And the spindle have got a vertical orientation, or it can be seen as vertically placed from superficial to deep. And it is presenting itself in a deeper layers, not like stellate cells, Martinotti and pyramidal cells, which can be seen on the superficial layers. Another type of cell which can be seen on the superficial aspect is the horizontal cell. By the name denotes, the cell body is placed horizontally. It is also spindle shaped structure which have got a horizontal orientation and presenting itself on the superficial layer. And this processes as axons and dendrites, they are connected towards the pyramidal cells. They are not passing towards the white matter connections. So these are the different types of neurons that can be seen in a cerebral cortex. They are pyramidal cells, stellate cells, Martinotti cells, the fusiform cells, and the horizontal cells. This was the diagram showing the afferents and efferents of cerebellum. Actually, it was misplaced. Like that. This is how I kept here, because this is how appears a cerebellum with its outer molecular layer, intermediate Purkinje cell layer, and inner granular layer, like that. This is the cells, how it is appearing in the cerebrum with the different types of cells. The cerebral cortex. Cerebral cortex also shows different layers, like molecular layer. The molecular layer is the outermost layer that can be seen as a molecular layer. Then the next layer is outer granular layer. Outer granular layer. Next layer is the pyramidal cell layer. Then inner granular layer. Then inner ganglionic layer. Then multiform layer. These are the layers which can be seen: molecular, outer granular, pyramidal cell layer, inner granular, ganglionic cell layer, and multiform layer. These are the layers. I will tell you what all the things are present in these layers. So in